Thank you. Thank you, Deborah, and thanks to everyone who's here. Just a little bit of background about me so you know where I'm coming from. Before I came to IARC 6 years ago, I was an occupational epidemiologist for nearly 20 years in the US at the National Institute for Occupational Safety and Health, studying mainly cancer and occupational exposures to a wide variety of different agents. So I first want to say I have no competing interests to declare. And to give you a little sense of where we're headed, I'll first give you some background on the IARC Monographs program. Then I'll talk in detail about how we do the evidence synthesis and integration across different evidence streams. I will talk about the new priorities for evaluation in the monographs, and I encourage people to pick up a poster sitting in the back, which I'll show you, but it'll be much easier to see if you've got it in your hands. And then conclude with some future directions for both epidemiological and toxicological evidence evaluation. So IARC, for those of you who are unfamiliar with my agency, really consists of two strands. And I know you all are very familiar with DNA. So one strand generates data from interdisciplinary research, including a lot of genomics research. The second strand evaluates evidence through independent expert review. And it's this latter part of IARC's mission that I'll talk about today. So I am situated within the evidence synthesis and classification branch at IARC. We are one of 7 research branches. And this program contains the WHO Classification of Tumors program, on which all of the cancer diagnoses are based worldwide. It also contains the IARC handbooks of cancer prevention. And I know some of you have read those or even been involved in working on a handbook. And then the program that I'll talk about, the IARC Monographs on the identification of carcinogenic hazards to humans. This has been around since 1971, so we're into our 53rd year at present. We are fondly known, or maybe not so fondly known by some, as the WHO's Encyclopedia of Carcinogens. So our raison d'etre is to actually identify what can cause cancer in humans. And over this 53-year history, we've evaluated more than 1,040 agents. Why is this important? Why is cancer hazard identification important? Well, we know that there's a large global burden of cancer and that we're in the middle of an epidemiologic transition. We're going to see at least a 40% increase in cancer cases globally over the next 20 or so years. Why is this burden increasing? Well, we know that the populations are aging globally. This has a large influence on the number of cancers we'll see, but there's also been a shift in carcinogenic exposures to low and middle income countries. We also know from experience globally that prevention is the single most effective way to reduce that burden of cancer. And the first step in preventing cancer is to identify what causes cancer, which is the remit of the IARC monographs, and also what prevents cancer. And that's the work of the IARC Handbooks program. So we have a website you can visit, monographs.iarc.who.int. And this has all and everything that I'm going to be presenting today and more, including 134 published volumes of the IARC monographs. So the monographs aren't just an evidence synthesis program. That role is very important. We evaluate the results of the latest scientific research across a range of disciplines. But in addition, the monographs evaluations identify research gaps and thereby stimulate new research to fill those gaps. So as one example of how this works, in the 10-year period before 2007, we had about 140 publications on shift work and cancer. In the 10-year period after 2007, that number grew by 6-fold. It's not coincidental that the monographs evaluation of the carcinogenicity of night shift work happened in 2007. You can see clearly that these references are citing that evaluation and trying to fill gaps identified in that evaluation. As another example, more recently, in 2015, IARC evaluated glyphosate, which is one of the world's most commonly used herbicides, and there was a large increase in publications as a result of that evaluation. So you're probably curious to know how we do these evaluations. And since our inception, the process of converting the voluminous literature, which is depicted here, into our really iconic orange-hued volumes has been described in the preamble to the IARC Monographs. So I'll show you an image of the very first preamble published in volume 1 in 1971. And it gives all of the procedures for evaluating carcinogenic agents. So it's been published in the beginning of each volume since the beginning, but it's been updated periodically to account for changes in the science of evidence synthesis. The most recent change was made in 2018. This is when we convened an advisory group to revise the preamble. It hadn't been revised since 2006, and of course, a lot had changed since then, particularly in systematic review methodologies, but also scientific advances in cancer epidemiology and in carcinogen mechanisms in particular. So here is a depiction of the basic process by which we use. It's a 5-step systematic review process. Step 1 is identifying relevant studies. Step 2 is screening, selecting, and organizing the literature. Step 3 is evaluating study quality. And step 4 is reporting on study characteristics. And then step 5 is the evidence synthesis step. So the preamble gives the procedural guidelines for how all of this works. And in addition, tells us how to select participants for the meeting, how to avoid and manage conflicts of interest, how to involve stakeholders in the process, and how the meetings should be conducted. It also gives the different criteria that are used that I'll go through next to review the human cancer, animal cancer, and the mechanistic evidence. And then it gives a decision process for reaching an overall evaluation. The highlights of the new preamble include calls for increased transparency of the expert judgment that's used, increased rigor of the systematic review, and modernized methods for tools for searching and screening and documenting the literature. The monographs evaluations are done by working groups of experts. These are independent scientists who are deeply familiar with the scientific literature related to the exposure to the agent under evaluation and one of the endpoints that is being studied. It could also include methodological experts. It is these scientific experts who review the science and develop the evaluations. They're assisted in this by we in the Secretariat at IARC. We are scientists in our own right, and we coordinate all aspects of the evaluation and ensure that the preamble is adhered to. We may also have some other categories of participants who are there really for transparency reasons or for a very specific technical knowledge that they have, but in no case may these participants contribute to the evaluation of the carcinogenicity. We've got very broad global representation across our 50-year history of working group participants. Since 1971, probably now closer to 1,500 scientists from nearly 60 countries have participated in monographs meetings. And at these meetings, they consider 3 streams of evidence. The first is cancer in humans, the second is cancer in experimental animals, and the third is the mechanistic evidence. All these streams are integrated in reaching an overall evaluation. In addition, exposure is characterized in humans, and there's a group that also critically reviews the exposure assessment quality in the human studies, both for human cancer and human mechanistic evidence. So how are these streams evaluated? Cancer in humans has had the same basic principles for at least the last 40 years. And how it goes is this. For each type of cancer, there are 4 possible descriptors: sufficient, limited, inadequate, and ESLC, which means evidence suggesting lack of carcinogenicity. So one of these descriptors is applied to each cancer type. You know, there are hundreds of cancer types that could be eligible. How is that done? For its sufficient determination, the bar is rather high. The working group judges that a causal relation has been established. And what this generally means is that within the body of evidence, a positive association is observed, and they judge that chance, bias, and confounding can all be ruled out with reasonable confidence. Not absolute certainty, but reasonable confidence. For a limited call, They determined that a causal interpretation is credible. For example, there is a positive association. However, chance bias and/or confounding could not be ruled out with reasonable confidence. For an inadequate determination, the studies permit no conclusion about a causal association, or as is often the case, there simply are no data available. For ESLC, there are high-quality studies covering the full range of exposure that are consistent in not showing a positive association at any level of exposure. And this is an important point because the Monographs program does not randomly evaluate agents. To get onto the review by the IARC working group, there needs to be some evidence of carcinogenicity, either for cancer in humans, cancer in animals, or mechanistic evidence. So this is why the bar for ESLC is quite high. And what is often seen is that evidence suggesting lack of carcinogenicity occurs for one cancer site, but for another cancer site, there may be sufficient or limited evidence. For example, tamoxifen, which is protective for breast cancer, but causes uterine cancer. That's just one example. The next stream is cancer in experimental animals. And here, these are generally experimental designs. And so while the 4 same descriptors are used, of course, the criteria for judging them are quite different from the observational data that we have for human cancer. So for sufficient evidence, there is a causal relationship established, and this means there are multiple positive findings for increases in tumors for animals that have been exposed experimentally, ideally in studies conducted under good laboratory practices. For limited evidence, the evidence is weaker, that's more suggestive of carcinogenicity, but there's only a single study or only an increase in benign tumors. And then there are also criteria for inadequate and ESLC. The mechanistic evidence is the stream that changed the most in the recent preamble. And it was really a result of a review that was done in 2010 of all of the group 1 carcinogens up to that time. I know I haven't told you yet what group 1 is, but group 1 means sufficient evidence, a known carcinogen is in group 1. And at the time of volume 100, there were about 100 agents that were classified in group 1, known carcinogens. So a group was convened to reevaluate all of these 100 or so agents, confirm which cancer sites had either sufficient or limited evidence, and then to explore what the mechanisms were for these known carcinogens. So as a result of this process, a workshop group helped to answer some of these questions, which were how to search more systematically for relevant mechanisms for carcinogenesis, how to bring more uniformity to the assessments that were being done. An increasingly problematic issue is how to analyze this voluminous literature that's developed for mechanistic evidence. And then, importantly, how to avoid bias towards favored mechanisms depending on who's in the room at the time. As a result of an evaluation of all of these group 1 agents, these workshops developed what we call now the 10 key characteristics of carcinogens. These are the chemical and biological properties of the established human carcinogens. This is very important. These are not hypothetical pathways to cancer, but these are the behaviors and properties of the known carcinogens. Data on the key characteristics can provide evidence of carcinogenicity, even in the absence of actual human tumor and animal tumor evidence. This is the principle behind the use of these mechanistic data. And they may be used to assemble the literature and the data on mechanisms without needing to have an a priori hypothesis of how that pathway would occur. So here listed on the left, you see the 10 key characteristics of carcinogens. These are a bit arbitrarily classified, no doubt about it. There could have been 15, there could have been 8, but in the end, they grouped them into these 10 categories. So for those of you who study cancer, many of these are quite familiar. And broadly speaking, they go roughly in order of a process that might occur, but not all of them necessarily occur for all carcinogens. Some types of carcinogens exhibit only one key characteristic, others have many of the key characteristics. And there is a literature that's available describing how these were developed, how they're used, and what endpoints can be used to measure the key characteristics. So this is a depiction here showing some of the agents that have been demonstrated to have these effects. So for example, electrophilicity and genotoxicity, We have many chemicals that exhibit this behavior. Key characteristic 7, immunosuppression. Many pharmaceuticals exhibit that behavior. Many viruses cause immortalization. And a lot of the particles and fibers are inducing chronic inflammation. Some of the persistent organic pollutants, like dioxins, modulate receptor-mediated effects. And some have 8 or more of the KCs. So once we have these key characteristics, how then do we evaluate the evidence? 3 descriptors are used here: strong, limited, and inadequate. And for strong, the evidence shows that the results are consistent across different systems. So you can look at these in systems like exposed humans, experimental animals, or in vitro systems. The overall database is coherent, and the studies cover a range of relevant endpoints in mammalian species in particular. The evidence is considered limited if the data are suggestive, but for example, the studies only cover a narrow range of experiments, endpoints, or species, or there's unexplained incoherence across studies that have similar designs or between different endpoints and test systems. We often have inadequate data, which is when there are few or no data available, or they are of highly questionable quality, or of course, if the results are negative. But increasingly as well, that it was recognized that there are different topics that influence the mechanistic evidence. The first, of course, is what I've just described, the key characteristics of carcinogens. And these can be in exposed humans, in human primary cells or tissues, or in purely experimental systems. And these are thought to be ranked in terms of their relevance to humans, with exposed human mechanistic data thought to be the most relevant. There could also be a determination that the agent belongs to what we call a mechanistic class. And this is a class of agents that have already been classified in group 1 or in group 2A, probably carcinogenic or carcinogenic to humans. And the agents behave very similarly mechanistically. So this is a special determination that's made about whether the agent may belong to a mechanistic class. The last determination is whether the agent meets very specific criteria for saying that the mechanism seen in the experimental animals is not relevant to humans. This is invoked more rarely, but it's there in case we have strong evidence that that's the case. So I've walked you through the 5 steps. Step 5 happens at the 8-day meeting that we hold in Lyon, which is advertised on our website, about a year before the meeting takes place. And this is where the working groups synthesize the evidence within each evidence stream, and then they integrate the 3 streams of evidence together to reach the overall evaluation. So here we are. Group 1 means carcinogenic to humans, which now has 128 agents. Group 2A, probably carcinogenic with 95 agents. Group 2B, possibly carcinogenic with 323 agents. And the largest group, which is group 3, not classifiable as to its carcinogenicity to humans with 500 agents. Okay, how do we get there? How does this integration happen? Well, first of all, it happens in one single step. There used to be upgrades and downgrades, but now the mechanistic evidence stands on its own next to the other 2 streams. The human cancer evidence trumps all the other types of evidence in the sense that if there is sufficient evidence of carcinogenicity in humans, then the agent belongs to group 1. And that's been the case, as I said, for decades. So some examples of this recently include opium consumption, which was classified with sufficient evidence for cancers of larynx, lung, and bladder. It also had limited evidence for cancers of pharynx, esophagus, stomach, and pancreas, and it was classified in group 1. More recently, occupational exposure as a firefighter was evaluated and found to have sufficient evidence for 2 cancer types, mesothelioma and bladder cancer, and limited evidence for 5 more cancer sites. And this, again, was classified in group 1. Another way to reach group 1 is if we don't have sufficient evidence in humans, but we do have sufficient evidence in animal bioassays for causing cancer and strong evidence of the key characteristics in exposed humans. So this particular combination is another way to reach group 1. And we have a recent example of this. Are people familiar with PFAS compounds? Have you heard about that? Is that a big issue here in the UK? Probably. So a working group evaluated PFOA, which is one of the legacy PFAS compounds, found sufficient evidence for cancer in experimental animals and strong evidence in exposed humans for two of the key characteristics, epigenetic alterations and immunosuppression. There was also limited evidence in humans for cancers of the kidney and testis, and there was also additional strong evidence for the mechanistic evidence in experimental systems. So all in all, this then reached group 1. Group 2A is less than sufficient in humans, but it's a combination of two streams or more. So there could be limited evidence for cancer in humans and or sufficient evidence for animal cancer or strong mechanistic evidence. And some examples here include a solvent called 1,1,1-trichloroethane, which had limited evidence for multiple myeloma and sufficient evidence in animal bioassays, and night shift work, which had limited evidence for human cancer for several cancer sites, sufficient evidence in animal bioassays, and strong mechanistic evidence, but that evidence was not strong in exposed humans. So this was in group 2A. Another way to reach 2A, if the evidence is inadequate for human cancer, is the combination of sufficient evidence in experimental animals and strong mechanistic evidence in human primary cells or tissues. So this is more relevant to humans than purely experimental systems, and so that allows the 2A classification to be reached. Our advisory group felt that there needed to be some direct human relevance, which is why to reach 2A, there needs to be at least evidence in human cells or tissues. And so we had several chemicals and metals that were classified for that reason. Trivalent antimony was lit up the board here. We had all ways of reaching group 2A with trivalent antimony since it had limited evidence for lung cancer in humans. And then lastly, the strong mechanistic class that I mentioned earlier, if there is found to be that evidence, then that reaches to group 2A as well. So here we had aromatic amines and a single epoxide that were found to be behaving like other agents that were in group 2A or group 1. Group 2B is reached when we only have a single stream of evidence and it's less than sufficient in humans. So for example, limited evidence alone for cancer in humans. And here we had aspartame getting classified here for this reason. We had only limited evidence in humans for hepatocellular carcinoma. There was also limited animal bioassay evidence and limited mechanistic evidence, but the driving determinant here was the human cancer evidence. So group 2A or 2B. If there's only sufficient evidence for cancer in animals, again, that's a 2B, and we have many, many chemicals that fall into this category, which are the subject of experimental bioassays. And the last category, strong for mechanistic evidence alone, here we had just 3 compounds, including one of the PFAS compounds that we evaluated in November. Interestingly, if you have strong mechanistic evidence, but it's not in human cells or exposed humans, it doesn't get you into group 2A. You're still in group 2B. And we had a few examples here. And then lastly, all other situations not shown above, like limited evidence in animal bioassays, inadequate evidence for human cancer, this goes to group 3. And we have many, many agents in that group. So I wanted to point out that there have been many evaluations that have included mechanistic evidence in humans. And these are generally endpoints related to oxidative stress, chronic inflammation, receptor-mediated effects like night shift work. But most of the time, this evidence is found to be inconclusive because it's either inconsistent, there are few studies, or the quality of the studies is not great. And I know that there's a lot of work going on in looking at mechanistic endpoints in this group, looking at pathways to cancer using MR methodology. We haven't seen that kind of evidence really in the studies that we've looked at to date. And perhaps that's due to the nature of the agents that we've been evaluating, which include a lot of things like chemicals, which are all shown here, heavy metals, complex exposures like firefighting exposure. Now here we did end up having strong human mechanistic evidence because there were a lot of well-conducted studies in firefighters that demonstrated some of the key characteristics that are listed here. So PFOA and PFOS, as I mentioned, had strong evidence. And for PFOA, that was very influential in putting it into group 1. Some of the future agents that are on that chart that you see do have mechanistic evidence in humans, but it remains to be seen whether those will lead to a strong determination or not. And I don't want to go through all of them. So we're beginning to talk with the research community about how to make mechanistic studies more informative for the monographs evaluations. We definitely think the choice of study population has a lot to do with it. If we have a high exposure contrast, it's much easier to conduct a study where you don't need a huge sample size. Having adequate power, minimal selection bias and confounding, as in any observational study, these are important considerations. And sometimes this is given less attention in these types of sort of panel studies or cross-sectional studies that we see. In addition, the endpoints that are selected are also very important. And it's difficult to measure some endpoints like chronic inflammation. Often we have acute measures of inflammation repeated over time. And when does that become chronic inflammation? The quality of the exposure assessment and simply the quantity of the studies are all concerns at the moment. So one of the important features of the monographs is that there's a lot of attention paid to two things: study quality, which includes an evaluation of the quality of the exposure assessment and the cancer outcome assessment, potential for selection bias, consideration of adequate control for confounding, the quality of the data collection methods, and the appropriateness of the statistical analysis methods. There's also consideration of what we call study informativeness. And I think sometimes in the intervention world, that might be considered directness of the study, to answer the question of relevance for the examination. So it goes beyond considerations of study power and can consider the presence of an adequate and meaningful exposure contrast, the adequacy of the latency and any relevant time windows between exposure and susceptibility to getting cancer later, as well as exposure to the target organ. All of these would be important determinants of the informativeness of the study. When synthesizing the body of evidence, the monographs uses an adaptation of the time-honored or not Bradford Hill viewpoints. And they have a lot of caveats. So the strength of association is definitely important, but weak associations may be causal. And for many of the things that are on a high priority list where there's widespread exposure, we do anticipate the metrics, the strength of association will probably not be strong. Consistency is important. However, that has to account for differences in study quality and study informativeness. And there's a lot of attention paid to this as you read the evidence synthesis for each monograph. The presence of an exposure response is said to be, a very strong indicator of causality. However, we know that confounders can also exhibit exposure response patterns. So this is not necessarily alone enough. The temporality of association is required, and that can be problematic, especially in some of the biomarker studies where it's collected after the diagnosis or even the treatment of the disease. Clearly, that's a concern. And then biological plausibility. And here we don't consider the mechanistic evidence to contribute directly to the human cancer synthesis because it's considered as its own evidence stream. Rather, here they're looking at really exposure of the target organ, latency, and that sort of thing. So one thing that's changed with our new preamble is that we have now incorporated a very detailed evidence synthesis section, which incorporates the working group's assessment of the roles of chance, bias, and confounding about their conclusions on causality. It also contains evaluations that are specific to each cancer site, and it gives explicit documentation of the expert judgment and rationale of the working group in reaching its conclusion. So one might ask, how robust are these expert judgments? We had an opportunity to look at this in the Volume 100 series 15 years ago when all the 100 Group 1 agents were reevaluated by different working groups. In fact, all of these agents were reaffirmed as Group 1, and for many, the evidence had strengthened since the previous evaluation. Why was that? I think it has something to do with the quality of the studies and in particular, the improvements in exposure assessment that were contributing to more less biased estimates than we had seen previously. In addition, there's been broad concordance between the findings of the monographs program and the conclusions of other hazard identification programs that use different approaches than we do. For example, they don't convene expert judgment or expert bodies to use expert judgment. Well, what about agents that have inadequate evidence? Is that a stable state? Back in 1985, well, we have an opportunity to look at this because roughly 40 years ago, there was a workshop convened partially by IARC and partially by Oxford University to discuss the interpretations of the so-called negative evidence in human cancer studies for 10 agents, again, not picked at random, but that had sufficient evidence in animal bioassays. So these you could consider having a strong prior of being carcinogenic coming in. This group concluded that for most agents, their judgment was that they thought the evidence would be likely to remain inadequate or even suggesting lack of carcinogenicity indefinitely into the future. But in subsequent years, 3 out of the 10 agents were found to have sufficient evidence in humans, and 4 out of the 10 had limited evidence. So most of the agents moved beyond inadequate evidence. And you can show and document that it was improvements in the epidemiologic study number, quality, and informativeness that were key to these changes. So we think that sufficient evidence has been a fairly steady state, whereas inadequate is often just a lack of good high-quality studies for agents that have strong evidence from other streams of carcinogenicity. So how are we doing? I'm doing okay. I want to talk about priorities, but before I do that, I'd like to advertise our little human cancer chart here, which we know as Homo monographiensis. This is a depiction of the cancer types that have sufficient evidence for different agents. So you can download this poster or look at it on our website. The ones in red are the agents that have sufficient evidence of a causal effect, positive causal effect, and the ones in green have a sufficient evidence of a preventive effect. And you can see there's a lot of disparity across the cancer sites in terms of what we know about their causes. With lung cancer, I realize it's a little bit hidden by the graphic here, but lung cancer has by far the most known causes. Even though we know tobacco smoking is responsible for most lung cancers, in the absence of tobacco smoking, there would still be many, many known causes of cancer, lung cancer. But for other types of cancer, we know very little. Breast cancer, is quite surprising that we only have 6 agents that have sufficient evidence for breast cancer, which is kind of shocking to me. Okay, let's look at the priorities, and this is the poster that you have access to in the back. These were recommended by an advisory group that we convened in March of this year. And just to briefly orient you, this yellow circle in the middle are the agents recommended for high priority evaluation. And there are roughly 120 in this group. Our group is very small. We cannot do all 120 of these. So we are going in and sort of prioritizing them ourselves. The color indicates the evidence that's contributing to the nomination. So light blue is human cancer alone. Purple is human cancer combined with mechanistic evidence, and green is human cancer combined with animal cancer evidence. So you see here, if you just look at the gestalt of this, a lot of purples, which means a lot of human cancer and mechanistic evidence in the pharmaceutical arena. And we'll go through these in diving deeper into the different quadrants. So the advisory group is depicted here. These are scientists, nearly 30 scientists from 22 countries with no conflicts of interest. And this summary was published online in the Lancet Oncology, and you can find it again on our website. So taking the top left quadrant, here are a lot of persistent organic pollutants in the circled area. These tend to have a lot of animal bioassay and mechanistic evidence. Next to that, we have miscellaneous chemicals, which have a lot of mechanistic evidence and some animal bioassay, but very little human cancer evidence. And then we have the physical agents, which are a bit of a mixed bag with some human cancer evidence, as well as the other streams. Artificial light at night is interesting. That's one of those agents that's linked to night shift work. And that has evidence, it's indicated in black, which means all 3 streams were contributing to that nomination. Up in the upper right quadrant, we have a group of solvents, some of which do have human cancer evidence. A large number of pesticides, which have a lot of mechanistic evidence. Some have human cancer evidence as well as animal bioassay evidence. And this is an interesting group. And then here we have dye-related agents, including hair straightening products, which is for a first-time appearance in the monographs, as is tattoos and permanent makeup, which were given medium priority. A group of metals, particles, and fibers, including complex exposures like air pollution. Outdoor air pollution is already in group 1, but now the interest in the community is about identifying the components of air pollution and which cancers are related to them. There's some evidence, for example, that nitrogen dioxide may cause different cancers than particulate matter in air pollution. So this is interesting in another way because things that emit pollution, like gas stoves, tend to emit a lot of nitrogen dioxide. So it would be nice to know if that's carcinogenic. Down here, we have a lot of the biological agents, including several viruses and several parasites that can cause liver cancer. And then a lot of pharmaceutical agents. Like the pesticides, we had a huge growth in this group, and these are based largely on the combination of mechanistic evidence and human cancer evidence. So we would anticipate doing quite a number of these in the next 5 years or so. And then the last group are what we call the complex exposures. So I think this slide really encapsulates a lot of the areas of research of the group here in Bristol, with some things like sedentary behavior, electronic nicotine delivery systems, sedentary behavior, and even cannabis smoking, perhaps. So how can you get involved if you're interested? There are a number of ways to do this. You can respond to our calls for experts, which we publish a year ahead of the meeting. So we're getting ready in a week or so to announce the meeting that will be held in June of 2025. So it will be a set of agents from that list that I showed you previously. Maybe things you're interested in, maybe not. I can't tell you what they are yet, but it's probably a week away from posting that. So check out our website. We also have a newsletter you can subscribe to get these kinds of information. You can also respond to the call for data, and you can consider the publication of our priorities list to be a call for studies on these agents. I know I've talked with Richard and Sarah the last day or so, and there's a lot of research going on here. So we encourage you, if you are publishing work in these areas, to publish your studies. The data do have to be publicly available. Usually that means in the peer-reviewed scientific literature, but also it can include studies that are reported on government websites. Often we have animal bioassays that don't make it into the published peer-reviewed literature that we find this way. All right, future directions. I've got about 6 minutes to get through this, and I'll try to speed through it. We've had our advisory group on priorities. We are now planning our monographs meetings for the next couple of years. We're also working on a database that I'll show you soon. It's called IARC Mondo, and we're trying to make the relevant information from our monographs more broadly accessible to people without having to dive into these hundreds of pages of the books to get the information. That's the idea behind Mondo. We also want to improve and update the tools that we're using, particularly some of this high throughput data that's being used to evaluate the mechanistic evidence and integration with omics data. So some of the things I've discussed today, or the last couple of days, would be really relevant here, particularly studies in exposed humans, since they're so valuable in our program. We're planning for a scientific workshop on research gaps for high priority agents that have unresolved carcinogenicity. We want to integrate the IARC Mondo database with the GloboCan database of IARC, which maps out all of the cancer rates around the world, and I'm sure many of you are familiar with that. We're very interested in the relevance, the mechanistic relevance of lipid metabolism and glucose imbalance. And again, we've talked about that and the overlap with the work here. And we have a plan to reevaluate all the group 1 agents to identify new cancer sites, but we're seeking funding for that major project. The mechanistic evidence continues to evolve. Last summer, our toxicology group convened a workshop of experts on mechanistic studies. And the themes of the workshop were to help with increasing quality of the interpretation of the endpoints that form the mechanistic evidence base, incorporation of this high throughput and omics data into the KC's construct, integrating all the different key characteristics themselves into the mechanistic evidence evaluation. All those were the main themes. And you'll be seeing some publications on that. So I'm going to skip the Mondo details that you don't need to really see right now, probably. But Mondo will tell us things like, what are the causes? What kinds of things cause different types of cancers? So for example, this is lung cancer. As I showed you, we have many things that cause lung cancer. 46 agents have either sufficient or limited evidence for lung cancer. And over half of these are particles or fibers, unsurprisingly, PAH related, which might surprise us, and metals. And then the rest are a mix of all these other things. But the pattern is quite different for bladder cancer, for example. We have fewer of these, but half of these are from occupational exposure circumstances or dyes or related chemicals. So this tells us sort of, we call these meta evaluations of the monographs information. And we want to do more of this. So the research recommendations workshop, we're aiming to hold in 2026. And this is a follow-up to a workshop we held 15 years ago, where we tried to identify major research gaps that needed to be filled for these unresolved agents. And so we published an update from the 2010-2009 workshop, and that will be the launch for the next workshop, where we're going to pick about 20 different agents with unresolved carcinogenicity and bring together experts who are doing different kinds of research to identify the major gaps that need to be filled. And again, I think this is an area of potential collaboration. So where are we going with the human cancer evidence evaluation? Clearly, having sufficient findings of sufficient evidence in humans are important, because alone that can lead to a group 1 evaluation. So we want to, clearly have a look at how we're making these conclusions. We want to continue stimulating research for the group 2 and 3 agents whose carcinogenicity is unresolved. We're interested in, we do apply meta-analyses where warranted and continue to do so. Publishing quantitative exposure response information where they're supported by data can be helpful as well. And we would like to better apply AI tools to help with data extraction. And I think this is an area that many systematic review groups are exploring right now. It might be nice to join forces and see if there are tools that are useful. We also have a forthcoming scientific publication on methods to evaluate the impacts of bias and confounding. So why is this important? Well, it's fundamental to the evaluation of the human cancer evidence and the human mechanistic evidence. There is also a lack, generally speaking, of methods to formally evaluate the direction and magnitude of biases for cancer hazard identification. And we think there has been somewhat problematic focus on algorithmic scoring approaches, like GRADE in particular, where observational studies are just given a very low score. They start from an area of deep suspicion and they can sort of claw their way back to respectability. That's not the viewpoint of the monographs, obviously, because we've relied on these studies for 53 years. But still, we have a lot of challenges in evidence synthesis. And I'm showing here a few questions from working groups that actually occurred during the meetings. Okay, could selection bias that was caused by differential participation of the cases and controls have completely explained an odds ratio of 2 in this key study? Is confounding by co-exposure to arsenic likely to completely explain the positive findings that we see for antimony and lung cancer? Could smoking confounding completely explain the elevated risk of bladder cancer in these key studies? What's the relative importance of non-differential measurement error versus recall bias in this set of studies? So how do working groups get at these types of difficult questions? Well, one way is to explicitly evaluate the exposure assessment quality and to anticipate the impact on the measures of association. Not so much the magnitude of the risk, but whether the association is still observed. Evidence triangulation has always been a key part of the monographs, although we have not used that word as much as perhaps we could. But we always evaluate all available evidence. So no matter what the exposure condition is, the working group will evaluate it. And if we see positive findings from studies that have different types of bias, that can provide strong evidence for a causal role. Using causal diagrams to help elucidate confounders versus effect modifiers can be very helpful to working groups so that they're speaking the same language and considering what would be the important set of confounders to have adjusted for. Recognizing the trade-offs of different study designs to control for bias. Mendelian randomization is a great tool, but it's often not available for the kinds of things that we're studying. And it is one study design among several possible ones. I think our working groups often use negative control outcomes and exposures. These are outcomes plausibly related to the confounder, but not to the agent. And even if the authors themselves of the original study didn't specifically identify a negative control outcome, it's often possible to find one in the cohort study in particular and to use that. And then when we don't have good confounding information, sometimes working groups do use indirect adjustments for confounders, or even theoretical adjustment based on worst case assumptions about the distributions of exposures and confounders. So we've convened a year and a half ago a scientific workshop and are nearly ready to publish the scientific publication with the aims of helping our working groups by developing a toolkit of epidemiological and statistical methods to assess the impacts, the direction and magnitude of bias in the published literature. So this is geared towards the reviewer rather than the practitioner. It carries out worked examples throughout the book and has a definite focus on hazard identification and not risk assessment. We're using the Breslow and Day classical volumes 1 and 2 of the Statistical Methods in Cancer Research as our models. And this will then be the 5th volume in the Statistical Methods series. And we see here a title. The editors are Amy Barrington de Gonzalez, who's at the Institute of Cancer Research, David Richardson from University of California, Irvine, and myself. And we have 2 people in this room who participated in the workshop, Deborah and Sarah. So thank you so much for that. This is a brief outline of what the book will show us. We talk about the motivation for this work. It's founding in cancer hazard identification, use of DAGs and graphical tools to assess bias, and then chapters on all of the major sources of bias. Chapter 6, very excitingly, talks about how to incorporate bias assessments into evidence synthesis. What do you do when you have not a lot of data? What do you do when you have more data? And then chapter 7, which is geared towards the people who have access to the original data. And this is a plea to the research community to use more of these tools in their own research, which would really facilitate the work of the reviewer in evidence synthesis. The 4 examples that we use throughout the book are directly from the monographs, and the yellow highlight means that the evidence was limited at best for the agent in any 1 or more cancer sites, and for opium, we had some sites with sufficient evidence. So that's the books. It's coming out. We're probably a month away from that being published, but we do plan a major rollout when that happens, and it will be freely available on our website. So I want to close with another colorful graph, this time showing the agents that we've evaluated since volume 100. Only the environmental agents, because I did this for something for the NCI. And what we show here is in the middle, these are all the group 1 agents that we've identified since volume 100. In the next circle out is group 2A. Then we see group 2B, which has a whole bunch of agents, and then group 3 out on the edge. And again, the color scheme here clues us into what was the evidence contributing to the evaluation. And the point I want to make here is that even though you only require sufficient evidence of cancer in humans to reach group 1, it almost never happens that that's the only source of evidence that's convincing. So all of these ones that are sort of multicolored means that the evidence is coming from all 3 streams: human cancer, cancer in animals, and mechanistic evidence. There are a few instances where that's not the case. We've had, I think PFOA is on this list now, which does not have sufficient evidence in humans, and a couple of chemicals where that's not the case. So it's quite rare for us to actually make a conclusion that is only based on human cancer evidence. But the mechanistic evidence in particular helps really increase the confidence in the findings of our evaluations. So in conclusion, we think that cancer hazard identification remains important for cancer prevention. We've heard it said that all of the causes of cancer have already been found. I don't know if you have been hearing that as well, but some people at IARC have been saying that. And we would posit that there are still a lot of unresolved carcinogens out there. We think the Monographs has a 50-year history as a scientifically rigorous and trusted source of cancer hazard identification information, and we want to keep it that way, which is why we need to keep improving our methods. So we continue to evolve these methods with scientific advances in epidemiology and toxicology in particular. And I will close by acknowledging our grant funding, which comes from the US National Cancer Institute for the last 40 plus years, and then the European Commission for nearly 40 years, and the US NIEHS as well. And you see here a picture of our small but mighty team in the monographs, and probably half the people in this picture are visitors, visiting scientists or students. So we only have about 8 or 9 scientists in our group, and a few support people. So with that, I would like to thank you for your attention, and I'd be happy to entertain any questions.
Thank you, that's great, thank you. Questions from the audience or from people online? Not sure how I get to see people online.
Yeah, it can be.
Busy room.
Stop sharing that.
Yeah, stop sharing.
Yeah.
Okay, Sarah and then Richard.
Can I just ask, in your cases, your key characteristics, one of them was that the agent should be metabolically active. Presumably that's not sufficient to say that it's potentially carcinogenic, because you then got the step from the metabolic activation of the cancer, that specific type of activation.
Yes, that's a great question. So there are a couple of the cases that in practice have found to not be able to stand on their own to provide strong evidence. And one of them is KC1, metabolically activated or can be electrophilic or can be metabolically activated to an electrophile. And that's because there's not enough strong evidence that that KC alone can cause cancer. So that one has to be seen in combination with something else. And usually that's with genotoxicity. So yeah, that one's good. And the other one that can't stand on its own is induces oxidative stress, because that was felt to be too non-specific to rely on solely. But many things act together, including oxidative stress. And so it's often seen in combination with other cases. Richard, good question.
Yeah, thank you very much. That was a great talk, really, really clear. I was just wondering, so with the ones identifying unknown risk factors, so because Paul Brennan wrote with George about if you compare international trends, then it suggests that 80% of cancers are potentially preventable in high-income countries. How do you select? Because then, if you're a small group, how would you kind of then that seems to me quite a difficult thing to do, is to then try and identify what could potentially be unknown and then select that for review with a kind of low probability maybe with the current evidence of providing success?
Yeah, I mean, that's a tough question. And it's important to know that we collect nominations from all countries. And some of the things that are on our list really are not prevalent exposures in high-income countries. Things like opium consumption, it's going to be a 0% of the burden in the Western world, but it's a pretty high burden, maybe 30% of lung cancers in Iran. So, causality is local, I guess is one of the conclusions. And some of these burden estimates are quite squishy on the exposure numbers in countries where the prevalent exposure is really occurring. So that's one thing. The other thing is that we know a lot more about the causes overall of lung cancer than we do for ovarian or uterine cancer. We know almost nothing about the causes. So this idea that we've somehow explained 80% of the causes is, I think, a little premature. But then again, as populations hopefully lessen their smoking rates, we're probably going to see the emergence of new lung carcinogens that have been a bit shadowed by smoking. Because for many of the agents that we know cause cancer in addition to smoking, we have enough evidence that people can look at the interaction between smoking and other exposure. In my own practice, radon exposure is a good example. High-level radon exposure, we've got good smoking data, and you can see there's a submultiplicative but greater than additive interaction between those two exposures. But if you don't look at that closely, you could just put it all down to smoking and say, oh, this is, and some jurisdictions do that. They say, if you're a smoker, nothing else could have caused your lung cancer. That was it. But we're already beginning to see this phenomenon with more cancers being diagnosed, particularly in women who've never smoked. And the people want to know why that is. Well, we've got other known causes of lung cancer, and we probably are missing some. So the percentage of cancers depends on the exposure entirely.
I'll just follow on from that, because when you were showing the number of carcinogens for each different area and said there's so few for breast cancer, and that's worrying, and my first response was yes, and then I thought, well, actually no, not if the ones that are known about actually explain a very large proportion. So if BRCA, a kind of Mendelian genetic cause, together with reproductive hormones explained, which I assume are on that list, I couldn't see that, explained 80%.
Yeah. That's a good point. Yeah,
it's not a problem that there's fewer than lung cancer.
Yeah, that's true. Not all cancers are equally likely to be exogenously caused. And the Monographs program does not look at endogenous causes like endogenous hormones, genetic factors. We don't look at genetic risk factors. So we look at gene environment interactions, but not. So you're absolutely right that we could know a huge percent, the cause of a huge percentage of the causes of cancer. But on the other hand, how much do we really know about what the actual agent was, right? So we know parity, reproductive risk factors are strongly predictive, but do we exactly know why? Is it directly due to that effect, or is that a surrogate for something else, right? So
I hadn't realised IARC doesn't look at endogenous.
No. Yeah, we don't. Yeah. So we are missing a lot of things on that list that we have the external hormones, but not endogenous hormones.
Richard.
But then the endogenous is part of the mechanism though, isn't it?
Effects on endogenous hormones.
Yes.
The external, that's what I was trying to get at, I think, is that these things that we understand to be endogenous might also have an exogenous component, right? And that would be fair game. It's part of the cases. Yeah.
Just I noticed on the poster of all the agents of like carcinogens and also the things which help to prevent cancer.
Yes.
There are a lot of like absence of excess body fatness was listed as a lot of like the thing.
Prevention.
Yeah. But obesity is not really causing cancer.
You have picked up on a really important gap. Yes. So the long story is, well, I won't tell you the long story. The short story is the monographs were advised to look at obesity, but during a reboot of the handbooks program, they decided to do a handbook instead. So they looked at the obesity data, but they inverted the question and said absence of excess body fatness is preventive of cancer. So it wasn't ideal. And the problem with it is also they didn't look at the mechanistic evidence the same way we would do in a monograph. So as a result, 2 months ago, our advisory group said, hey, we're putting obesity back on your list. Do it this time. And we've been already talking about doing it. And there's a lot of interest in us going to that to do it. So nowadays, the handbooks program, I think, is much more clear about what its goals are. And Sarah, you were part of the advisory group that helped to advise us on this. But instead of just looking at traditional observational exposure response information, now they're focusing on changes and intervention. So the most recent handbook was on reduction of alcohol consumption and cancer risk. So they looked explicitly only at the studies of reducing exposure, whether that led to a drop in risk. And the evidence was sparser for sure, but that they made the determinations based on that evidence. And then step 2 then is, now that we know that reduction in alcohol exposure prevents at least a couple cancer types, what evidence is that policies to reduce alcohol consumption are effective at doing that? So it's a bit of a 2-step process now for the prevention program.
So maybe just go, because we were talking about it before this seminar, you could say a little bit more about the visiting scientists and the opportunities there. I guess largely for people who are working in the cancer area, but for also for people who are interested in methods and evidence synthesis, the options to spend varying amounts of time at IARC as a visiting scientist, both at early career and later career.
Yeah, thank you, Deborah. That's great. There are opportunities, and sometimes it does depend on funding. It's always easier if you have a little funding to support coming here, and people who are on sabbatical will definitely generally have that. Right now, we have a couple of visiting scientists, senior visiting scientists from the University of Minnesota, and one of them is helping us with a bias assessment for a forthcoming monographs meeting. So it's possible to come and work on a specific topic. Like if you see us announcing a meeting coming up and you are doing a lot of research in this area and want to join the Secretariat for a short period of time, there are ways to do that as a visiting scientist. If you're earlier career, or if you've got, more than 10 years or so of professional experience, you can come as a senior visiting scientist. And there's actually an IARC program for short-term stays that's targeting people from low and middle income countries. So if that's relevant, that would also be an option. So we encourage people to come. We're very open and want people to come and visit us for short-term stays, long-term stays, or to come and work with us permanently.
And as well as the evidence synthesis, it's clear from what you mentioned at the start and from work that several people here have done with Paul's group and others, that there's a big genetic component as well, and then DNA randomization component.
And topics related to metabolism, food, dietary exposures. Right now, there's a meeting that Paul has convened looking at unknown causes of renal cancer. They found this genetic marker in, you probably are familiar with the study that was published a couple months ago that looked at a very broad population. We're looking at tumor mutagraphs project, looking at mutagenic signatures on tumors, and found that 80% of the Japanese population has an unexplained tumor signature. And so there's a lot of interest now in using this data to design more traditional epidemiological studies to help understand what that exposure might be for renal cancer. But it could explain a large burden of the cancer there if it's causal. So these kinds of opportunities also exist to work in the laboratory groups.
Well, thank you very much. For those of you who are here, Mary's going to be around for another 40-45 minutes. I'm happy to talk with anybody. Thanks again, both for the meetings you've had previously and for your talk.
Thank you, everybody.